Good afternoon, everybody. Well, good morning. All right, I'm gonna start. Welcome to the first PENS Outstanding Women in Neuroendocrinology lectures. We had a really good turnout, so thank you, everybody, for attending. I'm Victor Navarro. I'm an associate professor at Harvard Medical School and a current president of PENS, and it's my pleasure today to open up this event. So today marks the beginning of what we hope will become an annual tradition, celebrating the contributions of exceptional women in our field and honoring an active scientist for her outstanding work in a specific area of neuroendocrinology. This year, we're focusing on reproductive neuroendocrinology. It's a field that's seen a remarkable advancement thanks to the tireless efforts of many brilliant researchers, many of whom were women, and have overcome significant challenges in their careers. So we're honored to present the Neena Schwartz Lecture in Reproductive Neuroendocrinology. Neena Schwartz, who passed away in 2018, was a true pioneer in reproductive biology. Her groundbreaking work on ovarian function and hormonal regulation has shaped our understanding of reproductive processes. But beyond her scientific contributions, Dr. Schwartz was a tireless advocate for women in science, co-founding the Association for Women in Science and becoming the first woman president of the Society for the Study of Reproduction. So today we're privileged to honor Professor A. Terasawa from the University of Wisconsin-Madison for her lifelong contributions to reproductive neuroendocrinology as the speaker and honoree of this Neena Schwartz lecture. A will deliver a talk entitled Living Through History of Reproductive Neuroendocrinology, Research on the Hypothalamus in Rodents and Non-Human Primates. So this is a rare opportunity to have a glance into the lifelong contribution and in the career of somebody like A. Terasawa. So today we're in for a treat, but it's actually a double treat because we're fortunate to have Professor John Levine, who is the director of the Wisconsin National Primate Research Center at the University of Wisconsin-Madison, who will introduce this lecture. And John is really the perfect person to introduce this lecture because he's got firsthand knowledge both of Neena and A. So before we begin, I want to remind everyone that at the end of the lecture, we'll have a Q&A session. So use your Q&A box and we'll address the questions, as I said, at the end. And without any further ado, let us begin this inaugural lecture. Professor Levine, the floor is yours.
Okay. Oops. Let me. Okay. Can everybody see that? Am I?
Okay, you have to launch the presentation.
Yeah. Yep. All right.
Perfect.
Are we good? Are we good? Okay. I'm so grateful to have the opportunity to give an introduction to the lectures, both the lecture series today, the Neena B. Schwartz Lecture in Reproductive Neuroendocrinology, and an introduction to today's speaker, illustrious speaker, Dr. A. Terasawa, since I've had the great privilege to have been a close colleague and friend to both of these pioneering neuroendocrinologists. I first met Neena Schwartz at a symposium on reproductive neuroendocrinology held in 1983 at the then named Oregon Regional Primate Research Center, where I was a postdoctoral fellow. It turned out to be a meeting that really changed my life. Neena sat me down and grilled me about my work and ideas for the future, and thereafter recruited me to join the Department of Neurobiology and Physiology at Northwestern. So preparing for my interview, I did a deep dive on Neena's scientific record and quickly realized that this very forceful and a brilliant person that I had been talking to was responsible for a major share of the basic understanding of the neuroendocrine regulation of the reproductive axis and more, and much, much more. And she did much of that in the 50s and the 60s when huge barriers still existed for women pursuing careers in endocrinology and the nascent field of neuroendocrinology. Neena completed her PhD in physiology at Northwestern in 1953 and began her career as an assistant professor at the University of Illinois College of Medicine in 1961, serving as professor, assistant dean of faculty affairs, and professor of neuroendocrinology. In 1973, she joined the faculty of Northwestern University Medical School. And the following year, she was named as a Deering Professor and Chair of the Department of Biological Sciences at the Weinberg College of Arts and Sciences. In 1980, she founded Northwestern's program for reproductive research, now called the Center for Reproductive Science, which became a premier training site for students and investigators in reproductive endocrinology. So, during these years, she made seminal contributions towards understanding the hypothalamic pituitary gonadal axis and control, its control. Her science was from the beginning flawlessly designed and executed to allow for rigorous tests of her hypotheses. This was the characteristic of her science. Her early studies on the rat estrus cycle using newly developed, at that time, radioimmunoassays for pituitary hormone, she really established the basic tenets of cyclical changes in gonadotropin secretion on which our modern understanding of the axis are based. And this included the clear demonstration of a secondary FSH surge on estrus morning in rats. And she pursued this an understanding of this differential regulation of LH and FSH, and it led to her discovery of a non-steroidal feedback factor, inhibin, from the ovary involved in controlling the secondary FSH surge. And that is, of course, she originally wanted to call it folliculostatin. I think she regrets that she lost out and that it became inhibin. But her laboratory went on to make many important discoveries about ovarian inhibin. She received numerous awards and honors, including the Carl Hartman Award from the Society for the Study of Reproduction. She was elected to the National Academy of Arts and Sciences. She was also president of American Women in Science National Governing Board, and she really co-founded the National Association for Women in Science, and also within the Endocrine Society, founded Women in Endocrinology. She served also as president of the Society for the Study of Reproduction, as president of the Endocrine Society. And above all, Neena had the vision, drive, strength, collegiality, and professional integrity become a leader in the field and at Northwestern. Her legacy at Northwestern includes the continuing success of the Center for Reproductive Science, where she started in the 80s, and soon thereafter included young faculty such as Fred Turek, Mary Hunziker-Dunn, Kelly Mayo, myself, eventually Teresa Woodruff, who later became director of the center. So here's a little flavor of what was going on in the 1970s related to the public interest and understanding of reproductive neuroendocrinology. Here is an interview that Neena took at Northwestern and was asked to respond to the question, are women really unfit for leadership positions because of behavior which may accompany raging hormone imbalances? Her response is classic Neena. Quote, both men and women experience shifts in hormone levels from hour to hour, day to day, and during the life of cycle of birth to old age. There's no more evidence that these shifts limit the capacity of women to work well and feel well than there is that they limit the capacity of males. There was also a lot of work at the time that she was recognized for, and of course they called her Wonder Woman, on birth control and how it may be new strategies possibly engendered by discovery of inhibin. So Neena published her autobiography, A Lab of My Own, in 2010. She used this very personal memoir to come out as lesbian and encourage other women and gay scientists to persevere and succeed in their scientific endeavors. Here's one quote. I was the only woman getting my PhD in the Department of Physiology at Northwestern. At my first job at the University of Illinois, the chairman of the department asked me to pour the tea. Sure, I said, but maybe someone else can pour it next time. Nobody ever asked me again. So here's a group picture from that 1983 symposium that I referred to earlier. First thing you will notice is that there were exactly 3 females in the group. One was Berta Scharrer, who was the first person to propose that neurons can have this strange function of neurosecretion, which of course was a founding concept in the field of neuroendocrinology. The 2 other women were, you guessed it, Neena Schwartz and A. Terasawa. FYI, we have Ernie Nobile here. We have Doug Foster. We have Roger Gorski. We have Bill Crowley. Let's see. We have Reed Norman and Don McCann. They were all there to argue about the control of the primate menstrual cycle, with Neena moderating to ensure that they didn't kill each other. And we had Dr. A. Terasawa there, the small person who became a towering figure in neuroendocrinology, presenting her work on GnRH in monkeys. So leads me to today's speaker, who is A, and who's going to tell us about, in her own words, about her scientific journey through these early days of neuroendocrinology to the present. Dr. Terasawa is a professor of pediatrics in the School of Medicine and Public Health at the University of Wisconsin-Madison, and a senior scientist here at the Wisconsin National Primate Research Center. She earned her PhD in medicinal physiology at Yokohama University Medical School in 1966, and then as a postdoc, she joined the laboratory of Charles Sawyer at UCLA, where she took part in some of Sawyer's studies that established the concept that hypothalamic clock governs the reproductive axis, a fundamental tenet of the new field of neuroendocrinology at the time. In 1980, the WNPRC and the University of Wisconsin was lucky enough to have recruited A to the center, directed by Bob Goy at the time. It's here that she embarked on a remarkable career in the study of the reproductive neuroendocrinology of non-human primates. She has, from the earliest days, brought her, I would say, creative spirit, her rigorous scientific approach, her technical fearlessness, and limitless energy and endurance to develop a basic understanding of the neuroendocrine control of puberty, menstrual cyclicity, feedback regulation of GnRH release, GnRH neuronal development, neuroestrogens and GnRH release, metabolic influences on reproductive hormone secretions, and on and on. So I also have to say that she's the first one here in the morning and the last one to leave at night still. Please welcome Dr. Terasawa, who will be giving our lecture today on living through history of reproductive neuroendocrinology, research on the hypothalamus in rodents and non-human primates. And let me stop sharing here. And actually, let me switch over. Okay. All right. Okay.
Okay.
And you can advance here.
So, thank you for kind introduction. And I'm quite nervous about this presentation, but nevertheless, I hope you get some history of neuroendocrinology. So my mentor is mentor three. Masazumi Kawakami. He was graduate education responsible, and Paula Stimulus and Charles Sawyer, people called Tom Sawyer, was a mentor for my postdoc. And then after I got a job in the University of Wisconsin, Bob Goy, it was quite a bit influential to my professional life. So Dr. Neena Schwartz, she was a pioneer of the reproductive neuroendocrinology. I met her in a conference in the fall of 1967, that a small conference in Santa Fe Spring, California. And because of the women scientists were not many at that time, I was paired with her about the same hotel room, and I was absolutely devastated because I'm just a young starting scientist, and she's already quite a well-known scientist. But anyway, that's my big memory. Well, anyway, I'll go to the my work. So earlier stage of my career, I did some work about laboratory rodents. 1975 to 1980 approximately. So that time, we didn't know the anterior part of the hypothalamus preoptic area is responsible for cyclic ovulation, but it's anterior preoptic area, hypothalamus is quite a big area of the brain. And so I had a graduate student when I came here, and he and we decided to have a more exact spot of the brain. Is important. So we made a very much meticulously small lesions. So then we found that the ABPB, that the name is changed. When we called MPO, the medial proleptic nucleus, and it's a small nucleus of here, right above optic chiasma. Oops. I'm sorry, the preoptic area, and that is lesion. That induces the persistent stress. And it was reported that the suprachiasmatic nucleus, here you can see, it's a little bit more caudal area. The lesion here is already shown that also important for a cyclic ovulation because of this nucleus receives the neuron directly from optic tract. But when we make the lesion, this optic nucleus is completely intact. And so we did also the different animal. We made a lesion in the optic nucleus, and indeed we have we can get the persistent estrus, but this is because of disruption of the day-night cycle. And this is more than that. And eventually now we do know this nucleus has a lot of kisspeptin neurons. And the kisspeptin was not discovered at that time. And the kisspeptin neuron, as you probably everybody know here, is very much important in regulation of GnRH release. So then we moved into the guinea pig study. Because of this primate center had some colony of guinea pig. Guinea pig is, it's a rodent, but somehow they have a luteal face. And the short follicular phase, unlike primate, and few days of follicular phase, and then full luteal phase. So every 14 to 16 days, they do cycle. And those animals, we did the study. And that time, as I said, timing of the preoperative research depends on the time of day, importance of the environmental rights. That Everett and Sawyer, they described. But in the monkeys, Fred Cash, who was a postdoc that time with Nina Vilslav, they said monkey is really different. Nothing to do time of day. Actually, it's more depending on how much estrogen is given, how long does it take. Larger the dose, latency to elevate surge is shorter. If a smaller dose, latency to the surge is the longer. So we did the guinea pig, and because of that, we thought it's very interesting. So actually, I should more think guinea pig classification rodents or not has a little argument. I'm not going to go into that issue. But anyway, so the guinea pig is somewhere between rats and monkeys. So the dose of estrogen affect largely for latency, but also exactly the LH surge is very much locked in like a rodent. It's just time of day. That's what we discovered. But at that time, director of the Primate Center, Bob Goy told me, well, this is not rodent center, this is a primate center. So I had to switch to the monkey study. So what at that time what's going on is, as I said, Nina group, hypothalamus is important as a source of GnRH release, but not for preparatory LH surge. Estrogen action on the anterior pituitary is sufficient for induction of preparatory surge in the monkeys. And then Harold Spieth and Reed Norman in the Oregon Primate said, no, the GnRH surge is very important. And that time already we had the Sari and Gilman. They isolated the factor from hypothalamus released to stimulate LH and the first generate 10 amino acid peptide. That was the isolated and molecular structure was reported. So now you can measure you don't need to use always LH as an indicator of GnRH, and you can measure GnRH. So when I get into this monkey neuroendocrine field, I decided to have more directly measured regenerative release. And we used push-pull perfusion method or microdialysis method. That I'll just show you how we do it. But anyway, we directly measured. And also, my laboratory did some cell culture system of degenerate neurons. And earlier we did some embryonic nodes. That's where degenerate neuron originate from. We cut off the fetal nodes and then cultured, and we did lots of work. So then eventually we moved to make the embryonic and induced pluripotent cell to have a make the degenerate neurons. So this is, I said, this degenerate neuron originate from this olfactory placard. That's the place degenerate neurons are born, and then graduate all the way to the hypothalamus. So knowing this characteristic, we make the time-mated pregnancies. And embryonic 36 to 38, we dissect out this nasal area. And then the culture, you can see degenerate neuron. So we did use this model. And, but it's this time-mated fetus to make a degenerate neurons extremely important, extremely expensive. So we decide to make a degenerate neuron from pluripotent stem cell and embryonic stem cell from human. We decide to make a degenerate. And then this is, we already reported, but stem cell, we derived FGF8. Both human embryonic stem cell or induced the pluripotent cell derived from human skin, you can get the degenerate both. We have to prime the FGF8 during some differentiation process. And culture for 3 to 4 weeks. But this, because of the costs are so expensive, we decide to have stem cell, the neuron induced pluripotent stem cell and embryonic stem cell. We make the degenerate neuron. That's what we did. So this is just stem cell. We just make the neuron, but on the top of it, make a neuron, we have a FGF treatment, FGF, fibroblast growth factor 8. You can make the degenerate neuron, both embryonic stem cell or human-induced pluripotent stem, both of them. So now we don't need to make, when we do need a degenerate neuron, we don't need a time-mated fetus. So we did some cultured cells and make the chambers and artificial CSF slowly perfuse here. So then this perfusate, we measure the degenerate by radioimmunoassay. You can see that this degenerate neuron, they are active individually, but once in a while, 1, 2, 3, they synchronize and release degenerate in a pulsatile. Even degenerate themselves, they have a pulse generating mechanism that and we did hot chemicals stimulate or inhibit, that in vitro study we did. But of course, after experiment, we always confirm degenerate neurons in the slide. And now moving to the in vivo studies. primarily we did two different research. We tried to find mechanism of puberty onset in the primate. What stimulate the timing of puberty? I mean, what stimulate the puberty? That's a big question. And that we are still studying. I will talk to you a few minutes later. Then the next question is, we found that the brain makes estrogen. It's called neuroestrogen. That is very important in control of degenerated, how neuroestrogen play a role in degenerative disease. We are still studying. But anyway, microdialysis experiment, how we measure this pituitary stalk is here. This is a pituitary. This is a side view of monkey taken by X-ray. So we put the stainless steel pedestal, we implant initial surgery, and then the center of this pedestal is just the median eminence. That's where we aim. But anyway, so we can take the lots of sample from this base of the hypothalamus, and by perfusing the artificial CSF into the brain through the microdialysis of cannula, and then collect the sample to the fraction collectors. And then we measure this sample in a radio immunoassay or liquid chromatograph, the LCMS, MSMS mass spectrometry. But anyway, for the several different neurotransmitter, neuropeptide can go RIA, but some amino acid neurotransmitter, you have to use LSMS MS system. But anyway, now move into the main story that puberty, what triggers the puberty? And just briefly, I would like to introduce the pivotal stage. It's a monkey, female and male here. The degenerate neurons are quite active right after birth, and then somehow become very quiet period, and then start some sign of puberty, and then menarche occur, and some sparse menstrual cycle. But the first ovulation occurs about 3.5 years old. And the male is very similar, except for the first sign of puberty, externally it's hard to find, but if we take the blood sample morning, evening value is higher. Nocturnal LH increases very clear, become clear at the time of puberty, and then that's become higher and higher, and then eventually testicular volume increase and maturation of the testis makes spermatogenesis. It's male is slightly later than female, but essentially very similar in female to female. So this is LH changes throughout birth to the puberty. This is I borrowed that slide by Tony Plant's group, and as I said, LH release, or actually GnRH neurons are active first several months after birth, but then suppress. This is actually gonna detect now right after birth. Animals born. So that this is no gonadal inhibition of GnRH release. So the despite the fact that animals are gonadectomized, LH levels are really low, and then start to increase, and the females somewhere a little after, shortly before 2 years old, start to increase. And then towards the time of the puberty through ovulations, it's increased. Male is slightly delayed, but the same thing. So that based on this LH and some other external sign and the changes in developmental LH changes, we infantile, prepubertal, early pubertal, mid-pubertal, that's what we did lots of studies. So what neurotransmitter is important for initiating puberty? That's what we have been addressing for many, many years. And of course, as I said, pulsatile GnRH is very active at neonatal period, but pre-pubertal period, both frequency and amplitude very much suppressed. And then start to increase at the time of puberty. Frequency increase, but also amplitude slightly. And later time of development, frequency stays same, but amplitude gradually increase at the time until the time of ovulation. But anyway, kisspeptin is very similar to the GnRH. Neurokinin B is very similar. And glutamate also increase at the time puberty, GABA decrease. And neuropeptide Y is also somewhat similar to the GABA. So and we did some other substances, norepinephrine, neuropeptide, also that study, but I'm not going to go. Anyway, so big question what we have is, this is, I said, gonadectomized animal, or also gonadally intact animal, but it's even gonadectomized animal. Spontaneous GnRH release is become low during the prepubertal period. And so why? Of course, it's nature provide not to have a child bear the baby. So it just is positively inhibit. And then somehow this release from inhibition or just, we don't know the exact mechanism yet, but GnRH increase. It's just because of the disinhibition or GnRH neuron become more active. We don't know at this point. But anyway, so this is a big picture we do know, but exactly what this transition makes, what the signal is important. We are still studying. So now we have this period, prepubertal period, when GnRH neuron are suppressed, whatever. And I did quite a bit of work about GABA, but more recently, what we found is this naloxone, that opioid inhibitor, is, if we infused prepubertal period, huge increase, just before, after. And the pubertal period, naloxone, that the GnRH response to naloxone is, it's not no longer, it's completely lose. So somehow for us now, it looks like a naloxone opioid inhibition is very much important element of prepubertal inhibition. So what we are thinking is candy neuron, kisspeptin, neurokinin B, dynorphin, but anyway, those three neurotransmitters are very important for us. Cyclic release of GnRH, that was shown by Mike Lehman and Bob Goodman's group. And because of the sheep and rodents, they co-localize. But Eric Hanabowski shown that in the primate, those three neurotransmitters are not too many neurons co-localize. But so we think that the independent neurons still interact each other to regulate both kisspeptin, neurokinin B are stimulatory, but opioid neurons are inhibitory. That's what we think right now. So I just, last part of my talk, I want to talk about neuroestrogen integration of the. So this discovery of neuroestrogen is quite almost accidental because we are studying what estrogen causes GnRH release. So we had a cultured neuron and then give them cultured neuron under microscope, drop the estradiol benzoate, GnRH neuron become right away to be active. And so GnRH is excitatory to directly human GnRH, actually monkey GnRH. So that's what in that case maybe in vivo study, same thing we can find. So in a microdialysis experiment, we did this GnRH, measuring GnRH, and they gave estradiol benzoate, and here 10 nanomolar, and 10 times higher, 100 nanomolar. You can see as soon as we infuse estradiol benzoate into the stalk median MS area, estrogen release start to increase, really huge increase. And higher dose, maybe it's a little bit longer each pulses, but somehow estradiol benzoate stimulates GnRH release, GnRH and estradiol. So I condition behavioral control. And we confirmed that if we stimulate the basal hypothalamus, you can see that both LH and estrogen release. So it's estradiol benzoate is exciting some element in the hypothalamus. So in that case, what happened? Block the synthesis, local synthesis of letrozole. So that's what we are doing. This just under vehicular infusion. If you as I show you the estradiol benzoate stimulate the release of the black line and 100 nanomolar in this case, and right around the time of before, during, after, if we have a letrozole, block the letrozole, you can see no effect. So somehow this estradiol benzoate action is de novo, that de novo synthesis of local estrogen in the hypothalamus. That's what this data show. So we are quite surprised brain makes estrogen. And we studied quite extensively what the role of neuroestrogen on DNA release. And one of the things we have done is if we give the estrogen-induced LH surge, if we give the letrozole at the same time, it's attenuate. So obviously, estradiol stimulates pituitary LH, but the same time, this estradiol is stimulating GnRH. So if you block the synthesis, that's the lower. So, so that's a female. And we thought we should test the male because some people may not know brain hypothalamus in the rodents are sexually differentiated. In other words, after neonatal period, if testis is present, then mature male has a different type of hypothalamus as compared to female. But if you castrate at neonatal period, male brain retain the flexible flexibilities and respond. But that is not the case. In the monkeys, the hypothalamus, even neonatal period, the testosterone is around, the hypothalamus is not sexually differentiated. So we tested this role of neuroestrogen in the male and female. And this is the data is from male monkeys, group of male monkeys. You can see a control or a letrozole, essentially very similar surge occurred. So unlike female, the neuroestrogen is not involved in the male monkeys. But anyway, I just decided not to go. I talked so many things, so I'm not going to summarize. But I just want to, at this point, make a little commercial. How important research in non-human primate. And I'm sure there are some graduate students and postdoc in the audience. If you have an opportunity, you should get into the non-human research, neuroendocrinology in non-human research, because physiological processes is similar to those in the human, and experiment not possible. In the monkey study, you can use, like you cannot do the human, that you can do. And yet findings are directly applicable to human health. Of course, there are monkey research need more complicated animal protocol and quite a high cost, but nevertheless, it's very worth. And I just want to have some, this is not a complete list, trainees and some collaborators I just listed here. And over the years, I received many support from NIH. Thank you very much. Any questions?
Hey, thank you very much. It was an amazing presentation, Seb. Thank you for that, and congratulations on this amazing career that you've had. So anybody with questions, please use the Q&A box. I'll read most of the questions, or as many as I can, and also let some people in. While I do that, the first question that we had is from David Jaramillo. Does the structure of neuroestrogen vary at all from peripheral estrogen. How does one distinguish between the two?
Did you hear that? The question?
And
Can you repeat it?
Yes. Does the structure of neuroestrogen vary at all from peripheral estrogen? How does one distinguish between the two?
between?
Between neuroestrogens and peripheral estrogens.
Oh, so it's the same. It's same estradiol, no molecular differences. It's the same, but neuroestrogen is somehow independent sometimes, and sometimes helping the peripheral estrogen action. So it's a kind of surprise what we discovered. It just landing into neuroestrogen issue almost accidentally, as I explained to you. That's the case, and we just have to recognize. Of course, neuroestrogens are postulated in the learning. Like hippocampus is very much influenced in the presence of neuroestrogen, regardless of sexes, male, female. Both male and female makes neuroestrogen anyway. But we didn't know neuroestrogen also play a role in GnRH release.
Sounds good. I'm going to let the next person that want to make a comment or question in. I think he definitely deserves to be in. Hi, Tony. Can you see us? Tony?
Gotta unmute, Tony.
Is that better? Yeah.
There you go.
So it's good to see you, Victor and John. I'm glad to see you're still working hard, and so enthusiastic about your work. It's been a pleasure for me to listen and see you again.
Thank you. Any
Can
questions?
I ask a question?
Yes, of course.
The issue of the opiate as a brake. In our studies of the male, of the prepubertal monkey, this is the agonadal model, that however we gave naloxone and however much we gave of it, we never were able to demonstrate any evidence that there was an inhibitory opiate tone in the male. So
Yeah.
I wonder, have you looked at that in the agonadal female?
Yeah, so the yes, it's the same thing. The reason that you don't see is if you give the systemically naloxone, that go to stimulatory, inhibitory, both pathway in the hypothalamus. We are giving very local, just mini-aminase. That's what is the difference.
Okay.
Yeah.
Thank you.
All right, we have a few more questions in the chat. So the next one that we have is from Mike Lehman. Wonderful talk, Thank you so much for this and for all your fantastic work over the years. Your recent data on the effects of naloxone in prepubertal monkeys is really interesting and exciting. 2 questions. Have you examined effects of opioid receptor-specific antagonists like kappa, norbni yet? That's the first one.
We have been testing right now. Just, the naloxone is primarily mu receptor, but also cross-react with other, like a kappa and other receptors. So we are testing specific agonist.
And the second one that he has is, and have you looked at opioid antagonist effects during the luteal phase in adult animals, given the role of KNDy neurons in progesterone negative feedback mediated by dynorphin?
We have not done. We are all right now just a pivotal monkey. That's what we are comparing, not full adult animal. It's because it's too complicated.
All right. We have a few more questions here. Colin Saldana, he says, fantastic talk, as usual. Do you have any guesses as to where the neuroestrogen is synthesized? Where are the cell bodies?
Well, okay, so that's neuroestrogen is a neuron make it, but also some glia. That is known. But we have not looked at, but according to literature, some neuron and actually kisspeptin neuron has aromatase also. So kisspeptin neuron also may be a neuroestrogen synthesizing neuron. I think that's a very important question.
All right, we'll move on. We have a few more questions for you. Luke Ramachili, wonderful talk. I'm struck by how the neuroestrogen levels and GnRH levels are cycling so strongly and independently in the MB age after electrical or EV treatment. How do you think this cycling might work?
I'm sorry, I lost the question.
So the pulsatile release of estrogen after you electrically stimulate, how do you think that might work?
Well, I think that the electrical stimulation is, we put the electrode very close to the sampling. So it just, we are picking up the sample from media eminence arcuate nucleus region. It's more so media eminence. So locally similar, the cell components are stimulated. So probably some glia and some neuroterminal is stimulated. And so retrogradatory cell bodies are from media eminence that neurons are stimulated.
All right. Robin Bears asks, I really enjoyed the talk. Thank you. I was wondering what time the letrozole was given when you found that attenuated the LH surge in female rhesus monkeys.
So I don't get it. I'm sorry.
What time? What was the timing of the letrozole injection? How far before the surge?
Yes. So we are starting week before. And also this it's not because the luteal phase makes the complex because the progesterone comes in, which is we haven't studied, but we are just giving the gonadectomized animal, give the basic, very small dose of estrogen capsules. So high LH is generated released by gonadectomy suppressed, and then gives them the estradiol stimulation. I didn't talk, but progesterone, just a small dose of progesterone after the preoperative phase stimulate. And then luteal phase, that progesterone is inhibitory to the GnRH, but I decide not to talk about that area. It's too complicated right now, this whole seminar.
I guess, they want to know when you administered letrozole.
Yeah, so that's about 7 days before. So starting continuously, we are giving the daily injection of letrozole, yes.
Right. Andrea Gore asks, is neuroestrogen made in other brain regions than the hypothalamus?
Say again?
Is neuroestrogen made in other parts of the brain besides the hypothalamus?
as I said, the neuroestrogen is synthesized hippocampus and amygdala. So it is also this neuroestrogen is very important for like amygdala. And if it's neuroestrogen is low, apparently depressed. So estrogen is, as much as we know, estrogen is a happy hormone.
Mega Massa is asking, do you have a proposed mechanism by which, for why exogenous EB was unable to rescue the lack of neuroestrogens by letrozole?
I don't get it.
How do you have a mechanism by which estrogen, the estrogen that you're giving, I think she's asking, will act to counteract the effects of the letrozole? Is that what?
Oh,
is that?
Yeah. So no. So letrozole. So our purpose is letrozole is aromatase inhibitor. In other words, letrozole blocks the estrogen synthesis. But I didn't show the data. There's a very intriguing results we are getting. Letrozole works not only aromatase inhibitor, but more directly work as some estrogen receptor system. But it's complicated stories. I didn't talk about that.
All right, the last question that we have is from Denise Bolshan. She says, thanks, A, for such remarkable work over the years. You really were doing groundbreaking work before many others in the field. Monkeys are very expensive, and this is a barrier to their general use in most research labs. What advice can you give our younger colleagues that would like to use this model?
Well, NIH is they are willing to support monkey work if its aim is good and approach is good. So if you have opportunity, I encourage getting into the Primate Center complex, or like Tony Plant's that Pittsburgh had their own primate colonies. There's quite a few institutions had a primate colony, so is Harvard. But anyway, I think don't afraid to go into the primate research and then write a grant, good grant, and NIH will support you. I think NIH is trying to encourage scientists, but unfortunately right now, the primate neuroendocrinologists are gradually retiring and not young people are coming. So that's why I'm just really trying to solicit anybody who wish to study, just get into primate neuroendocrine area.
So if I could also do a little bit of advertising, that all of the 7 national primate research centers, including us, have pilot project programs where you can apply to do a pilot project with the centers in a way that will introduce you to the use of non-human primates, collaborate with insiders, and perhaps move your project from mouse or rat species to non-human primates in a way that will allow you to address some really translationally some very relevant model for translationally relevant problems.
Can I make a comment on primate use?
Absolutely.
I mean, I agree with A very much that if you want to understand the human menstrual cycle and human puberty, the non-human primate is theoretically the model of choice. But not only is it expensive, it's very difficult. And the beautiful techniques that can be applied to the mouse contemporarily involve transgenics. And of course, the rhesus monkey is really genetically intractable for most purposes. And I don't think that the monkey is a good model for a young scientist and young investigator, particularly graduate student, because it takes so long to study these processes. And I've thought about this a lot. How can one bridge, the supreme model of the monkey with the contemporary, beautiful contemporary methodology that you can apply to the mouse and the rat, I guess.
Yeah, well, I think that's limitation, but also I know the group of University of Wisconsin, they want to make a transgenic autism model. So it's a technology is a little bit behind, but I'm trying to help this PI to launch because somehow like autism, obviously there is a specific genes involved, and so it can be managed to see the monkey, how behavior changes, how interaction changes with other monkeys. But GNRH is relatively, now we do know more what the genetic is important for GNRH release and inhibition. And so I think that will lead, but I think that's an important area to aim future.
Yeah.
I'm sorry to have to stick my nose in here again, but as director of Primate Center, I've got to, Tony. We now have non-human primate studies that are using genomic engineering, genomic editing, single nucleus RNA-seq, ATAC-seq, gene targeting, and really it is a new generation of genetic, genomic, and molecular techniques that now can be used efficiently and directly in rhesus, in marmosets, in ways that are allowing for breakthroughs in questions that have not been able to that we've not been able to address in rodent species. So I just had to push back against that coming from the inside here.
I mean, that's good. That's really encouraging to hear, John. And I hope I'm around to see some of the results that you can get, particularly in the context of primate puberty, because I think that is going to be necessary to be able to manipulate gene expression and look at effects on the timing or the delay or the absence of puberty. It's still a fantastic mystery.
Yep.
Also, you said that about it takes a long time to make that go through the puberty, but like marmoset monkey is shorter, which I haven't done anything, and the rhesus monkey is longer, but it's shorter than human.
Yeah.
Humans.
I have not seen in the marmoset the time course of LH secretion in an agonadal marmoset from birth until the expected time of puberty. And I think unless you've got that non-gonadal inhibition, which is the break for the prepubertal period, then until you've established that in the marmoset, to me, it's not it hasn't been established as an appropriate model.
Before we leave, I have a question that I think the three of you are the perfect ones to answer. I recently had a discussion with a clinical colleague, and we, like rodent people, have always considered that the positive feedback of sex drives, so the pre-ovulatory electrodes, depends on the activation of KIS1 neurons in the AVPV. That's something that we've seen for the last maybe 20 years now. And this colleague was arguing that in the human, this positive feedback happens mostly at the level of the pituitary, that the hypothalamus is not that relevant. So my questions are, one, is this true? Have you seen this in the monkey? So does the monkey look more like the woman, or does it look more than like the mouse?
Turn off Tony's mic.
We could go on all night.
So, A, do you want to respond to that with the role of the pituitary versus the hypothalamus in the old Pittsburgh, Oregon duel?
Well, no, no.
Well, I think a lot of us, including, if I could speak for Tony, have accepted over the years that there's major regulation going on at the level of the pituitary gland, and that there are gene or H surges that occur. It's a question of whether they're necessary or not, and whether feedback can be elicited solely by the actions of estradiol on the pituitary gland. I think we called a truce many years ago, and we'll just leave it at that.
Yeah. I mean, for me, the best evidence for that is the human data with Coleman's and other hypothalamic deficient humans. If you give them pulses of GnRH that mimic physiological LH pulses, the females, these women will cycle and have surges. So the conclusion there is that you don't need a pre-ovulatory GnRH surge in the human, but I agree with John, in the monkey there is a pre-ovulatory GnRH surge. I don't think we know in the human.
All right, fantastic. Thank you so much. I think we can close this now. And again, I want to thank first amazing presentation. Thank you so much again for everything and for agreeing to participate in this first lecture of this series that we hope to repeat every year. Thank you so much, John, for taking the time to introduce the lecture and and thank you all very much for attending. We'll be in touch. So thank you.